All right, everybody, welcome. This is one of our webinars in our webinar series for Precisionary instruments. We're really excited to have you here, and we are thrilled to host Dr. Claudia Loebel. So Dr. Loebel is an MD, Ph.D., and she is an assistant professor of materials science and engineering, and also a biological sciences scholar at the University of Michigan. She attained her medical degree at the Martin Luther University Halle-Wittenberg in Germany and her PhD at ETH Zurich in Switzerland. She then completed a postdoc fellowship in the laboratory of Professor Jason Burdick at the University of Pennsylvania. And she was awarded the Pathway to Independence Award, also known as the K99, through the National Heart, Lung, and Blood Institute at NIH for her work on synthetic lung tissue models to look at mechanisms of alveolar epithelial cell dysfunction. So her research involves the development of precision cut lung slices, or PCLS, to investigate mechanisms of early epithelial cell differentiation during lung injury and fibrosis. And so today she's going to talk to us about PCLS with a focus on epithelial cells and its application in pulmonary fibrosis research. And she's going to also discuss the imaging and the quantification of PCL metrics in her research. And with that, Dr. Loebel, thank you so much for being with us, and I want to give you the microphone.
Thank you so much, Abby. It's really a pleasure to give this webinar here today. And I'm very thankful for that you asked me to give this webinar, because it's also an opportunity to show our research. And so thank you so much for the introduction. And so today I wanted to present a little bit, provide an overview of what my lab has been doing, and then dive a little bit deeper into the precision cut lung slices that we've been doing using the Precisionary compressed-dome. So my lab, we started in fall 2021, so it's just been 1.5 years, and we've grown since then quite a bit. So now we are currently 4 PhD students and a few undergraduate students. And so my lab is really working at the interface of materials engineering and mechanobiology, with a general focus on lung regeneration and repair, and in particular the epithelial cells, which I will dive into a little bit later. But I wanted to give this very brief overview because we are very excited, also based on my background, on polymeric hydrogel design. So we are not only applying these hydrogels, but we are really also focusing on the synthesis as well as the fabrication of these hydrogels. And hopefully in the end we can discuss a little bit further some of these works, because especially we are also using the compressed-dome for preparing these hydrogels for imaging and analysis. And then another focus of my lab is this interface between cells and the extracellular matrix, because when cells, organoids, and any kind of other cell types, when we put them in these engineered hydrogels, or even when we do precision cut lung slices, they keep producing extracellular matrix. So they're secreting proteins, and they're using these proteins for their function. And so my lab, although I'm not talking about it today, has developed a series of metabolic labeling and quantification tools to investigate this. And then this is what I'm going to focus on today, is that my lab has been focusing mostly on pulmonary diseases. So epithelial cells for in vitro, especially lung epithelial cells for in vitro models, as well as organoid engineering. And so one of the biggest questions we have is, what is the regenerative capacity of the adult lung? Yeah. And so when we're looking at these alveoli, so the tiny niche within the lung where the oxygen is taken up by the blood, we can see these beautiful niches that are surrounded by the capillary system. However, within patients that are started to have this fibrotic remodeling within their distal lung structures, we can see that these alveoli are losing their kind of their surface area, so less oxygen can be taken up by the blood. But we also see this really intense fibrotic remodeling, so the accumulation of extracellular matrix, which is also called this pulmonary fibrosis. And this is another image that images that I really like to show when we're at the left, we're looking at these healthy alveoli where we stain for collagen, which is part of the extracellular matrix. We can see this nice honeycomb structure of the alveoli with very thin fibers of extracellular matrix of collagen. However, in these fibrotic alveoli of patients, so these are mouse studies, but even in human patients, we can see that this interface is much, much stronger and much, much thicker, so less oxygen can be taken up in blood. So this was really a motivation for us to design our engineering approaches. And so to address this question, and what I'm going to talk about today is really these three pillars, or two pillars of my lab, which are using these hydrogels, these engineered hydrogels for the formation of organoids, so these three-dimensional complex structure, as well as using these precision cut lung slices that we've developed over the last year, or have started to adopt into my lab over the last year. And so I decided to also talk a little bit about these organoids because we use the compressed tone to prepare our hydrogels for analyses, as I mentioned earlier. So I was hoping that people in this audience would still be interested. And then in the second part, I'm gonna talk about our precision cut lung slices. And so when we think about organoids, and I think most of the people in the audience will know about the lung structures and have probably heard about organoids. So organoids are these beautiful three-dimensional structures, as we can see here, that arise from stem cell or progenitor cells. And then depending on the media, we can differentiate them into these more proximal areas of our lung, as well as the more distal, the alveolar regions of our lungs. And they have been very useful, similar to precision cut lung slices, for the development of drugs, testing of drugs, as well as better understanding development and disease mechanisms. And that's why we have these two pillars, organoids, as well as precision cut lung slices, to answer some of these questions. And so here again, we are really focusing on the distal lung, so the alveolar niche, because when we think about the niche, and this is a very naive view, we have epithelial cells, we have mesenchymal cells that are surrounded by the extracellular matrix. So in particular, what we are focusing on are these type 2 alveolar epithelial cells, or AD2 cells. And these cells receive paracrine signals from the mesenchyme underneath, or surrounding, as well as the extracellular matrix that surrounds both, or is underneath the epithelial cells, as well as surrounds the mesenchymal cells. And then in response to injury, which can happens every day when we're breathing in dust or any other kind of things that are flowing in the air, these type 2 alveolar progenitor cells, they can self-renew and they can differentiate into these more specific type 1 alveolar epithelial cells that are lining the alveoli and are responsible or allowing the gas exchange. But many of you might have heard that we can culture these type 2 alveolar epithelial cells. We can investigate them in using precision cut lung slices, as I will talk about later. But generally for the organoids, they always require, or have always required, matrigel, which is this complex animal-derived extracellular matrix protein mixture, which works very well for organoids. But it also has a very complex structure and composition, and it has a huge batch-to-batch variability. And so as an engineer, a few years ago, we really started to focus on identifying hydrogels that may allow us to mimic and replace this matrigel. And of course, we were not the first ones, and especially in other fields, people have been using these engineered hydrogel matrices for organoids, and in particular intestinal organoids. So where they've been using fibrin gels, agarose, or this completely synthetic polyethylene glycol hydrogel matrices. However, over time, although we can culture these organoids, they need to be relatively soft. So typically they're less than 1 kilopascal in the elastic modulus, which, especially when we are thinking about pulmonary fibrosis, this is way below the stiffness of the extracellular matrix composition within our lungs, even in healthy lungs where we have approximately 3 to 5 kilopascal. In addition, these organoids are pretty heterogeneous in size and structure, which may be beneficial for some of these studies. However, if we are thinking about downstream analyses, we often aim to have a more homogeneous size of, or the homogeneous size and structure of these organoids. And therefore, our design criteria for this project were that we wanted to develop a matrigel-free as well as a hydrogel matrix that allows us to form these organoids in a defined size and structure. And so here, the major components of this project were a hydrogel that is a hyaluronic acid-based hydrogel backbone, which is modified with groups that can be photopolymerized. In this case, these were Novonin groups. And I'm not going into the details of the chemistry, but the beauty of these hydrogels is that these are enzymatically degradable. So when we put cells inside, while cells are secreting enzymes such as MMPs, matrix metalloproteinases, they can break these bonds and can degrade the hydrogel. And then the other component were these human alveolar progenitor cells. In this case, we used some type 2 progenitor cells that were derived from iPSC cells. And these iPSC cells, they have a knock-in reporter for the surfactant protein C, which will come up through the entire presentation because this is one of the characteristic proteins of these type 2 progenitor cells, which is responsible for the production of surfactant protein, which maintains our lungs, the stability, so to reduce the surface tension. And for this project, and again, I just would like to highlight this a little bit and introduce people in this audience to this project, because I think it is something that this audience may be interested in. So in this case, we used our single cell suspension of these IED2s that were expanded first in matrigel, and then we embedded them into our synthetic hyaluronic acid hydrogels, with the idea being that over time, they develop these organoid-like structures. However, when we just embedded these single cells into a hyaluronic acid hydrogel, there was almost no organoid growth, as you can see on the left in this picture, as well as in this graph here. This is matrigel, and then just with our hyaluronic acid hydrogel, there was no organoid growth. However, when we just mixed in a very small percentage of matrigel, we could almost recover the organoid growth, very similar to our matrigel, 100% matrigel. But then we thought, given that matrigel, a major component is laminin, we decided to just physically mix our hyaluronic acid hydrogel with a laminin protein. And in fact, we see this increase and very successful formation of organoids within these laminin hyaluronic acid hydrogel composites. And of course, we also wanted to understand whether we can maintain this reporter expression. So this is the surfactant protein C reporter here shown in green. On the left, you see the matrigel, and on the right, you see this hydrogel with the laminin components. And in fact, we were able to maintain a very similar surfactant protein C, the reporter expression in both. And we also wanted to understand whether we can use these to maintain these cells, to expand and then passage our alveolar progenitor cells in these hydrogels using hyaluronidase. So this is an enzyme that breaks up our hydrogel. And in fact, over at least 3 passages, we were able to maintain the growth and the yield of cells within these organoids. So this was very successful in our opinion. However, the next question we asked, well, do we actually need 3D hydrogel matrices, right? Are they required for alveolar progenitor cell organoid assembly? And of course, we're not the first ones thinking about this, but in our case, we used a very simplified silicon mold or a technique. These silicon molds are just made from commercially available microwell tissue culture plastics that you can purchase from companies. And so we just made a negative mold. And then in a second step, we used our hyaluronic acid hydrogel precursors. We put them on top, and then we added a thiolated cover slip, which just means that allows this hydrogel to bond to our cover slips, and then flipped it into a normal culture dish. And so then we were able to use different silicon molds, and I hope you can appreciate that we were able to form these microwell hydrogels with a range of different sizes and structures, so ranging from approximately 200 microns up to 800 microns in diameter. And then after fabrication of these microwells, we seeded single cells, which were then just by gravity falling into these microwells, forming these organoid-like structures. And given that we were able to use different microwell sizes, we were then also able to form these alveolar organoids, Alveolospheres, with different sizes. And I just want to show this area here because it kind of depicts the homogeneous distribution of these organoids, which also had a relatively homogeneous size and structure. We also, again, made sure that these cells maintain their reporter expression, so the surfactant protein C as the reporter, as well as here in yellow, shown as the protein expression, the surfactant protein itself. And I wanted to highlight a little bit that we also dig a little deeper into the biology of this, because the surfactant protein C or B processing is a quite sophisticated process in these cells. And so we wanted to make sure that we were still maintaining this biology in our synthetic hydrogels. So when we are looking at these type 2 other progenitor cells, they're making the surfactant protein C and then store it in these lamellar bodies. And so we wanted to make sure that our cells are kind of going through this entire process. And first, in collaboration with our collaborators at the University of Pennsylvania, we first performed Western blotting to look at the surfactant protein B as well as the surfactant protein C. And in fact, when we compared to Madrigel and our synthetic hydrogels, there was a very similar expression of these proteins. And we also compared to these human type 2 progenitor cells that we received from donor patients, from donor lungs. So a very similar expression. But the other thing you can look at when you're investigating these type 2 other progenitor cells is that you can actually look at these lamellar bodies or lamellar inclusions. And so this is transmission electron microscopy of primary human tissue. So again, from human donor lungs. And you can see these beautiful lamellar bodies. And this is how the surfactant protein is stored. And when we looked at our Madrigel and our microwell cultured alveolar progenitor organoids, you can see very similar structures to these lamellar inclusions where the surfactant protein is stored within these cells. So in general, this was very successful in terms of showing that our gel system maintains the biology of these 82 type 2 alveolar progenitor cell organoids. So I think this showed that we have designed a Madrigel-free culture and expansion. But what I wanted to mention a little bit is these applications in disease modeling, where I'm now getting into where we actually used the compressome for analysis. And so I want to highlight two different applications. So the first one is when you think about these alveolar type progenitor cells, one application that we've been investigating is the transplantation back into injured lungs. And so this is in collaboration with Andy Van at the University of Pennsylvania. And in this case, we use these immune compromised mice, the SCID mice, and we use pleomycin administration. So most of you have probably noticed this. So this is for the what we use for inducing pulmonary fibrosis in these mice. In this case, we use pleomycin to induce an injury, so to kind of make space for our transplanted IAD2 alveolar progenitor cells for them to transplant. So we administer pleomycin, and then after 10 days, we dissociate our alveolar progenitor cell organoids into single cells, and we transplant them intranasally into our pleomycin treated mice. And then after 14 days, we use the compressome to make these 300 micron thick slices and then run immunofluorescence on these. And so in this case, what we first showed was in the matrigel, and hopefully you can appreciate that after 14 days, cells, so these human cells from our organoids are shown here in red, which is a human-specific marker, and blue is a marker for type 1 alveolar epithelial cells from the host tissue in this case. And so you can appreciate, hopefully, that these cells that we transplanted integrated into the host tissue. But of course, we also wanted to understand whether our synthetic hydrogel grown alveolar progenitor cell organoids actually perform very similar. And so in this case, we again made these organoids, and then we dissociated them and transplanted them back into our immunocompromised pleomycin treated mice. And so in this case, we were first looking at proliferation. Here we used a Ki-67 marker. And again, in both cases, these cells were able to integrate as well as maintain their proliferative capacity, as you can see by these arrows where we still found Ki-67, so proliferating cells after transplantation. We also looked at the differentiation of these cells. So here we looked at the surfactant protein C. In this case, we just used immunofluorescence of these proteins. And again, in both cases, as shown here in yellow, you can see that most of these cells, or at least some of these cells, maintain their surfactant protein C expression or production after transplantation. So, and this is just another picture that I really like to show, because you can see that, again, the cells here that we transplanted in red were integrating and were kind of forming these alveolar type structures. So these images we took after performing these precision cut lung slices. In this case, they were fixed, of course, but it was very helpful to make these thick slices, and I'm very happy to talk more about this in the end. So again, and I was hoping that this was an example for how we can employ these organoids for regenerative engineering. And then since I started my lab 1.5 years ago, we have now been using this platform mostly towards disease modeling. And I just wanted to highlight again this kind of transition in fibrotic remodeling. So when we look at the left here at the healthy alveoli, we have these relatively thin structures of the alveoli, but generally, when we look at the elastic modulus of healthy lungs, they range between 2 and 5 kilopascal. While in patients with fibrotic alveoli, first of all, you can see this thickening and stiffening of the alveolar walls. But generally, what we were most interested in is really that the stiffness of the lung, of this extracellular matrix, increases enormously, right? So at least 5 to 10 times compared to our healthy cells. But when you think about the organoids that I just showed you, they're limited to these soft hydrogels. So it is really difficult to form organoids within stiffer hydrogels. And so my student, one of my first students, Madeline, she took this challenge and said, well, I'm really interested in studying organoids in stiff versus soft hydrogels. And so she started doing this. So here we're looking at these organoids. So this is just a live dead staining. So live cells are in yellow, and the dead cells would show up in this purple pink. And so you can see that in matrigel, we see these organoids after single cells. However, when we're now going into 3D hydrogels with 4 kilopascal or 20 kilopascal in stiffness, most of these cells are dying. So this was not a valuable or a valid gel system to investigate these questions of stiffness. And so that's why she said, well, I think I can use these microwell hydrogel system to pre-aggregate our organoids, as you can see here in number 1. And then after 3 days, instead of using semi cells, we're embedding these aggregates as a suspension into our hyaluronic acid hydrogels. And so first of all, in matrigel, this of course works pretty well. You can see these huge organoids. But now again, in contrast, in our synthetic hyaluronic acid hydrogels, we are now also able to form these organoids that are embedded in 4 kilopascal or in a 20 kilopascal environment. And this now allows us to study how do these cells respond to these stiffening matrices. But the reason I show this is also because this might be of interest to this audience, because we use the compressome to image and visualize the cells within these organoids. Because one of the biggest challenges is that when you have these huge organoids, you can either make you can cryosection them, you can embed them for a microtome sectioning, but we are really interested in the three-dimensional structure. So we want to have some figure sections. And so we decided to use the compressome to make these sections. And so here, Madeline just colored, so put some dye into her hydrogel so that we can visualize it for this presentation. And then she uses the compressome, or she embeds them in agarose. So again, this is just a gel here to visualize it because the organoids we cannot see. And then after sectioning, she gets these thin sections depending on the size she's interested, which we can now use for immunofluorescence or for imaging. And so this has been working pretty well. And so here I'm just showing some representative images that Madeline took. So in this case, we're looking at organoids in Madrigel versus this 4 kilopascal hydrogel. And we're mostly interested in these epithelial cell markers, so these tight junctions, which are very characteristic, as well as equatorial junctions that you can see here in blue. And in both cases, you can see that they start to form these polarized structures in both the Madrigel as well as in the hyaluronic acid hydrogels, as well as the SO1, so the tight junctions that you can see on the bottom. So the compressome has really helped us to kind of move a step forward. Although these are just single C slices, we were now able to also get these more beautiful three-dimensional structures, which we weren't really able to get with our cryo-sectioned very thin slices. So this is the reason I show this here, because I think it's very useful. Okay, so I think this is what I wanted to show about the applications of our microgel hydrogels. And of course, these microgel hydrogels are also very modular in size and shape. So we can use different cell types. We've also used this for different organoid systems, but we can also incorporate cells or different extracellular matrix components into these hydrogels. And so in the second part of my presentation today, I will give an introduction to how we're using the compressome for our precision cut lung slices, and I'm happy to talk more about this. So when I started my lab, this was the first time we started to use this. So we've done a lot of troubleshooting in this case. And so I'm just bringing up this slide again, because we're again interested in this transition from the healthy, thin alveolar walls into the more fibrotic state of our fibrotic pulmonary fibrosis lungs, where we can see this thickening of the alveolar walls. And so this is work that is led by my other student, Donya, and she has really kind of taken this and developed the precision cut lung slices in my lab. And so when she started her PhD a couple of years ago, she said, well, yeah, I'm really interested in understanding what is happening in these alveolar regions in the distal lung. And so this is a nice picture depicting where you can see, again, it's a histology slide. You can see this nice alveolar structure and fibrotic regions, these fibrotic foci in human lungs. There's no gas exchange possible. And so she's also interested in this alveolar niche, as I mentioned earlier, where we have these type 2 alveolar progenitor cells and these alveolar type 1 cells that are responsible for the gas exchange. So looking at this differentiation from type 2 to type 1 cells, what she is interested in is that I mentioned earlier, during injury, we have these type 2 cells that are self-renewing and differentiating into type 1 cells. However, recently, like a few years ago in the field, or in general, we know that during fibrosis, this differentiation is limited, and we see this increase in these transitional cell states. And so all these cells, and this is what has been discovered over the last few years when we're looking at type 1 to type 1, type 2 to type 1 differentiation. All these cells are undergoing or going through this transitional cell state, which are these pre-AT1 transitional cells, or PET cells, how we call them. And so these cells, this transitional cell state is very transient. However, in fibrosis, what we have seen is that these pre-alveolar type 1 transitional cells will accumulate. And people have shown that in these fibrotic areas, so here we're looking at a precision cut lung slice where we stained for these PET cells, these transitional cells, using keratin 8, which is a very characteristic marker among others. And you can see these fibrotic foci are really enriched in these PET cells, in these transitional cells. So the hypothesis here is that, or generally what we've seen so far is that we see this PET accumulation in these fibrotic regions. But one of the questions is that we don't, although we've seen a lot of single cell RNA-seq and we know that these PET cells are there, we don't really know when and why these cells are rising during fibrosis. And so when she started her PhD, she realized that most of these studies that we've seen in the literature are really focusing on this late stage pulmonary fibrosis. So when we have this accumulation of PET cells and we have this accumulation of extracellular matrix. But these PET cells are really accumulating very early on. So when we have these, the early initiation of pulmonary fibrosis, which we still don't really understand what is going on. And so she said, well, there's currently not really a model of this early epithelial cell injury during fibrosis where we will see this initiation of PET cell accumulation. And so that's why she decided to develop a PCLS model that will help us to investigate how these epithelial cells are talking and communicating with their extracellular matrix, and what is the reason for this PET cell accumulation. And so in this case, we are using different mouse models, and I just want to give this one example where we use these stop mice that we purchased from that are available at Jackson. And so these have a excuse me, a constituous reporter for the surfactant protein C. And she isolates the lung by infiltrating them with agarose, which is very typical for preparing these precision cut lung slices. So we use 2 weight % agarose, infiltrate the lungs after euthanizing the mice. And then we use the compressed tome to make these 300 micron thick PCLS that we then culture in our media. And so this is how we perform it. So everything is done sterile, although we're still working in a semi-sterile environment. But the PBS, so this is the compressed tome, and this is the PBS that we collect our precision cut lung slices in, and this is the sterile PBS. But generally speaking, we have not had any contamination issues. And then I just wanted to show this, how we are making these slices and then collect them in the PBS, and then she's fishing them out and putting them into our dishes for culture. But going a little bit back in how this PCLS has enabled us to culture these alveolar epithelial cells. So first of all, we were able to maintain these type 2 alveolar epithelial cells. So these are things we've optimized over the last year. So in green, you can see the reporter of these type 2 cells. And this is at day 0, so right after slicing. But we were also happy to see that these type 1 alveolar epithelial cells, so here a marker for type 1 cells in cyan, that we were able to maintain this structure right after cutting these slices. But the key here is that we can investigate these transitional cells. And so when Donia started this, she first wanted to make sure, okay, all these cells are viable. So in this case, we just use live dead staining for these cells, and here's the viability in percentage. And she was also, which was very important to us, that these SPC positive cells, so these type 2 alveolar progenitor cells, maintained their expression and maintain to be viable, right? So in this case, you can see that there are approximately 10% of positive cells compared to the entire cell population, but and they're very rare, but in general, this was maintained over a culture period of 5 days. But more importantly, when she looked at these type 2 alveolar epithelial cells, which are cuboidal, right? She found that so at day 0, they are all rounded. This is what we would expect. But then after a few more days in culture, she found that these cells are starting to spread, yeah? And I should say that in this case, actually she has it here. So when we look at the surfactant protein C expressing cells, they're cuboidal, round shape, and then these cells are starting to spread while they're undergoing this transitional cell state before they become these very, very thin type 1 alveolar epithelial cells. So she got really excited that she sees these still SPC expressing cells that were starting to spread out. And her hypothesis was that these are these transitional cell states. And then she started to look at these PET cell markers. So in this case, we used keratin 8, which I mentioned earlier. And in fact, at day 0, so here again, you see in green the type 2 alveolar progenitor cells. And in this violet, you see the PET cells. There were very little cells at day 0 because they're very transient during normal regeneration. But then over time, you can see that more and more of these PET cells are emerging, but more importantly, you see this spread morphology. So that indicates that we're getting this accumulation of keratin 8 positive cells, PET cells, within our precision-cut lung slices. And then she just went on and wanted to, because there are many different markers of these transitional cells, and she just was interested in whether they're also starting to express some of these collagen markers, which are very typical for initiating fibrosis. So in this case, she looked at the procollagen, so before it's being secreted. And while at day 0, there's very little procollagen expression, over 5 days, you can see that, again, first of all, these cells are starting to spread, and they're expressing more of this PET cell marker. But she was very excited that she now also see this procollagen expression, which again, or like more and more confirms that these transitional cells are accumulating and starting to kind of help or like contribute to the fibrotic remodeling within these precision-cut lung slices. And so for the last part of this talk, I just wanted to go through some troubleshooting that we've done to develop this model, because currently it is working very well, but we had a few things that we went through that I thought might be helpful for this audience. So first of all, when we do these precision-cut lung slices, we were wondering whether we should embed them and slice them the day after euthanizing, like kind of after isolating the lungs, or whether we should culture them for 1 day before slicing them. Because this is what other people have done in the past as well. And so in this case, you're looking at live dead. So live cells are in green, the dead cells are in this purple, and hex is just a marker for the nuclei. And you can see that the viability, if we do the isolation and the slicing at the same day, we get a very high or pretty high viability. And I think this is after 2 days or 3 days in culture. While if we wait 1 day, we see this drop by approximately 10 to 15%. So to us, it is really important now that we isolate the lungs and cut them right away and not maintain them in culture before cutting. Another thing that we spent quite a lot of time on was the right media conditions. And although there's a lot in the literature, it's not very conclusive which media works best. And I would be very interested in hearing more opinions on that. But generally, we were interested in the base media, as well as should we add FBS, fetal bovine serum, or should we not add it? And so here we were comparing, this is just 1 day of culture, using the normal DMEM minus, these are all minus FBS for now, and then this DMEM F12, or the small airway epithelial cell media, because the hypothesis was if we want to maintain more of these type 2 other progenitor cells, maybe we should use this specific media. So first of all, if you look at the right-hand side, this graph here, you can see that there's a general drop over 3 days in the viability. But generally, the conclusion we made out of this data here, and this is a replicate of 3 different lungs, that we cannot really, I mean, the small airway epithelial cell media is actually performing worse. So just having normal DMEM seems to maintain the viability the best. And the next question was also, should we add FBS? And so in this case, again, we're just looking at HEX, so to look at all the cells and then the dead cells here on the bottom. And in fact, when you just look at these pictures, I hope you can already appreciate that the small airway epithelial cell media with FBS is not performing as well as the other ones. But generally, the conclusion from this slide is that with FBS, we see a higher viability over the time course of 5 days. And then this kind of drop in the small airway epithelial cell growth media is less pronounced. And I just wanted to show this slide. And this was a lot of troubleshooting that Donya did, but generally here we're comparing the normal DMEM with FBS, no FBS, and then we have this DMEM F12 without FBS and with FBS. And so the conclusion she made here is that adding FBS generally improves viability, and this DMEM F12 performs the best. And before I conclude, I also wanted to say that, of course, we did all this for these type 2 other progenitor cells. So her conclusion was that the amount of type 2 other progenitor cells that we can maintain is the highest in this DMEM F12 plus FBS. And I would love to hear other people's opinion about that. So generally, we believe that PCLS is really a powerful tool. And although we are still in the beginning of our studies, I think we have shown that PCLS may help us to have this early stage fibrosis model, because we're inducing this injury and we're seeing this accumulation of keratin 8 cells, which we are able to maintain in our culture conditions. And we're hoping to more investigate, or currently we're investigating what are the reasons and how these PET cells are communicating with their with the matrix and how much they're contributing to early stage fibrosis. And so, yeah, I would like to stop here, and I'm hoping to have a discussion about this data. And I really want to thank Precisionary. I mean, they've done a wonderful job in kind of getting us set up here and helping with any troubleshooting. And my current lab, as well as our collaborators at the University of Michigan, as well as at Penn and Charité in Berlin. So thank you so much.
Oh my gosh, thank you so much, Dr. Loebel. And actually, we have about 30 people with us today. And so if you want to, we do have some questions. If you want to unmute yourself and ask questions during the Q&A session, we definitely welcome that. I know that Ricardo has some questions, as well as it looks like Dr. Charlotte Dean. I am also happy to ask questions. Oh yes, Dr. Dean, go ahead.
Hi, hi Claudia. I really liked your talk. Thank you. So we've worked quite a bit with imaging lung slices as well, and looking at different cell types. And in general, what we find when we culture with serum is that we get a lot more fibroblasts in the tissue. So that we've also stuck with just having a plain media, as you showed. And so I just wondered if you've actually done a comparison to look at the cellular composition that you get in FBS versus, non-FBS cultures.
Thanks for asking this. So this is why I was hoping to do this webinar. So we've only focused on these SPC positive cells, and we see more SPC positive cells when we add FBS. And that is kind of, because I have other collaborators who also said we see more fibroblast accumulation when we add FBS, but our SPC positive cells are dropping without FBS, right? But we haven't really done a direct comparison. So I think we should do that. It's a good point. So what are you looking, like, what are you staining for when you look at fibroblasts? Because we have all, like, how do you identify them?
Yeah, I mean, usually the most successful thing we found is a fluorescently, is a sort of cell label based on vimentin. So it's kind of like a dye that you can buy. It's sort of like, it's not quite a conjugated antibody, but it's something along those lines, and it will just label all the fibroblasts in the PCLS, and that works the best. Actually, it has been quite challenging to find a good fibroblast marker that will work in the PCLS. But the sort of vimentin based stain is the one we found that works the best. So I'm very happy to, you know, give you the details about it. Yeah, no problem.
Thank you. Yeah, we should do that. Thank you for the question. We've really been focusing just on these FPC cells, which I more and more realize is not the right way to do. So thank you.
No problem. Thanks.
We just had a quick question about what concentration of FBS did you use, Dr. Loebel?
Yeah, we've tried a lot. I mean, generally use 10%. That's what we've been doing.
10%. All right. And then Dr. Azam Panche, do you want to unmute yourself? Here we go. To ask your question.
I can also read it because it's a great,
Yes.
Hi.
So, hi. Thank you very much for your great presentation. Could you please elaborate more about the increase of keratin 8 in PCLS? Is that mean that in normal condition, only the culture duration may induce fibrosis in this model?
Thank you for this question.
Okay, thank you.
This is a very important question because we were also surprised. So generally what we see so far is that just culture in PCLS will increase the amount of keratin 8 cells, which would mean that we're inducing this early stage fibrosis in these culture models. And I hesitate to make more conclusions about this because we're still investigating it. But I know that the amount of inflammatory signals will increase by just kind of cutting these tissues, right? And so I think we are inducing fibrosis, which in our hands just means that we, you know, this is the baseline that we have. We have this keratin 8 accumulation. But it's a great question because we are also a little worried about this.
Okay, thank you very much.
Great. We actually have a question from Dr. Yambo.
Hi, I wrote the question, but I can still ask. So in your data, mostly I see the longest day of experiment is day 5, right? Can I guess that the viability of your PCLS is 5 days? Have you done more than that? And I see you have done also with hydrogel and matrigel and PCLS. I saw some recent study, people have also combined PCLS with matrigel to extend the culture to 14 days. So have you done that? And because the study design is mostly for the early stage, you stop at 5. How long can you use those PCLS?
Yeah, that's a great question. I mean, we've mostly stayed until day 5. I mean, we've kept them for 14 days, but because viability drops, especially in these type 2 cells, we haven't really gone any further. But I did not hear about adding matrigel, so thank you for bringing this up. I think probably it will help by adding it to the media. So thank you so much for this question and bringing this up. We have not done it.
Just a little other question. Me, actually, I work on the physiological side, so it's very delicate. I saw you had few troubleshooting, like when I do for the asthma, for the airway contraction. So any little change in the condition might make my result really go strong. By looking in your data, you don't have much issue on the structure. if you do just that, you have any issue with that?
No, we don't. Yeah, we don't have so many issues. But we're not looking at any, which is probably not correct, right? Physiological difference. We're just very naively looking only on the cells and cell spreading, where we don't see huge differences. But you're bringing up a good point, because if if we're not careful enough, the downstream analysis we are doing may just still change, right, based on what you just said. So it's a good point.
And
thank you.
Last one, I see. I have a question. I think somebody asked something like that. The mouse you use, you induce fibrosis and then you prepare precision cut from these lungs?
No, we use healthy lungs for that. We just, the PCLS I showed from fibrotic mice were kind of just a validation that our markers and the PET cells are accumulation can show up. But no, these are all healthy lungs. And the mice are between 5 and 8 weeks.
Okay. So this transition from 81, 82 to KRT8 is just in normal lung, no fibrotic pro-fibrotic cytokine included in the culture?
No, it just happens, which is, interesting itself.
That's interesting. Thank you very much.
Thank you for the question.
Wonderful.
I see there's this question.
Ricardo asked,
factors.
Yep. Have you added fibrotic inducing growth factors to your PCLS?
We have not. We just cut and have looked at the PET cells. It's a good point, because perhaps we can have this, add some TGF beta, for example, to have another positive control. That's a good idea. We have not.
Great. Thank you so much. If there are no other questions, we're going to go ahead and wrap it up and end a little bit early. Dr. Loebel, again, thank you so much for your time and for sharing your research. We're all excited to be here about it. And everybody, thank you also for attending, and have a great week.
Thank you.